Podcast number 8. And here we're going to talk about how proteins are made in terms of how they figured out the codon table. And the codon table is what mRNA codon codes for what amino acid. So proteins are put together amino acid by ribosomes. That process is called translation. And translation, what the ribosome is doing is taking the carboxyl end of one amino acid and attaching it to the amino end of another amino acid, removing water. It's called a dehydration reaction. And the bond that is made is a covalent bond, a special type of covalent bond called a peptide bond. So it's a strong bond. And this is done by the ribosome, by the RNA part of the ribosome, which is both RNA and protein. And some experiments to try to figure out what ingredients in the cell were required to make protein. So they added DNA in the form of chromosomes. Chromosome is a DNA wrapped up in proteins, packaged in proteins. Energy in the form of ATP and GTP. We'll talk more about how to make these forms of energy later on in the course. But that's essentially the energy currency for the cell, so that's sort of like cash that the cell needs to spend in order to do stuff. Transfer RNA, you know about. Ribosomes, you know about. Messenger RNA, you know about. And amino acids, you know about. So they did an experiment where they added these things all together inside a test tube, and then used gel electrophoresis and looked for proteins. So gel electrophoresis we talked about when we talked about RNA, where molecules are separated in a gel, which is got holes in it, and big things get stuck on the top of the gel. Small things travel further toward the bottom of the gel, and you pull things through the gel with electricity. In the case of nucleic acids, all the nucleic acids are negatively charged because of that those 3 phosphate groups on the nucleic acid. In the case of proteins, it depends on the protein and what amino acids are on the protein. So usually when gel electrophoresis is done for proteins, they coat the proteins with a negatively charged soap called SDS, and then they pull them through the gel. And then they stain the proteins after the fact with a dye that binds to some side groups on amino acids. So when you see a dark band on a gel, a protein gel, that means that there's proteins there. Okay, so in this first experiment, they added all this stuff. That's what the plus means, is that was in the test tube. And when they do that, they get proteins being made in the test tube, and the proteins have a bunch of different sizes. Some are big toward the top of the gel, some are small toward the bottom of the gel. In the next experiment, they added all the stuff except for the DNA, the chromosomes. And again, they got proteins being made. And then in all the other experiments, if they eliminate energy but keep everything else, no proteins are produced. If they eliminate the transfer RNA, no proteins are produced. If they eliminate the ribosomes, no proteins are produced. If they eliminate the mRNAs, no proteins are produced. And if they eliminate the amino acids, no proteins are produced. So this is a pretty classic experimental design. They're trying to figure out what is required to make proteins. They have these 6 different components, and they eliminate one at a time to try to figure out which are required and which are not required. Okay? And from this experiment, we figured out that the only thing that's not required is DNA. So translation does not require DNA. It requires energy. It requires tRNA. It requires the ribosome. It requires mRNA and amino acids. In the last podcast where I went over the diagram that I drew, you saw all those things in there except for the energy. And the energy is used for the ribosome to move along the mRNA. The ribosome requires energy at several steps in order to move along the mRNA. Okay? So we're going to watch this video now. Let's see if I can tab out of here. And this video...
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production.
Okay, so this video is produced by Howard Hughes Medical Institute, and it uses animation models of what these molecules actually look like inside of a cell and tries to give you an idea of the scale of how things happen. Obviously, they're zooming in and out. But what you're seeing here is there's the nucleus, okay? And these little things are nuclear pores. And like on the diagram that we just went over in the last podcast, the mRNA or the DNA has been transcribed into mRNA. That mRNA has been processed, and now that mRNA, with the introns out and the exons spliced together, that mRNA is now coming out of the nuclear pore.
Of the particular protein that this gene codes for.
Now here's the mRNA. Here's the ribosome sitting on the mRNA, and it's walking along the mRNA. Notice that the ribosome didn't start at the very 5' end of the mRNA. The first codon is going to be 3' of that 5' end. It's not at the very end, but it is walking toward the 3' end of the mRNA here.
Several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life.
So the red here are each little dot is an amino acid, and the protein is folding up as it's coming out of the ribosome. Notice the ribosome is huge, and in fact, compared to the other things here. So these little green guys zipping around, those are transfer RNAs. The ribosome is so big that it takes about 70 amino acids to put together before you see any amino acids coming out of the ribosome. I also want to point out while we're paused here, this little red dot at the end of a tRNA is an amino acid. And later on you'll see as the tRNAs bring the amino acids to the ribosomes, first they have the amino acid, then it becomes part of the protein, and then they're empty in the E site and they get ejected.
Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape.
I think that's really funny. Computer tapes haven't existed since the 70s, so and obviously this was made since then, so I don't know why they said that.
The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA.
So why are they all zipping around giving you motion sickness in watching this video? They're trying to give you an idea of what is really happening in the cell. And in the cell, things are just zipping around and bumping into each other, and it's all chance. Whether or not the right tRNA bumps into the right site on the ribosome is just chance. And things are just zipping around.
The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain.
So if you notice, these guys that are leaving the ribosome don't have an amino acid attached, and the ones that are coming in do have an amino acid attached, and that amino acid is going to join this protein chain here.
As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
Okay, they don't show you the end, which is running into a stop codon that would separate the large and small subunits and releasing that protein. All right, so how did they figure out the codon table? All right, a couple of things before we get started. They knew that the codon table had that a codon had to be at least 3 nucleotides long, and that's because there's 4 different kinds of nucleotides in RNA. If it was 1 nucleotide per amino acid, then we would only have 4 kinds of amino acids, and scientists already knew that there were 20 kinds of amino acids, so 1 wasn't going to do it. If there are 2 kinds of, if there are 2 nucleotides per amino acid, then there would be 16 possible combinations. If you have 4 different types of nucleic acid, 4 to the second power, or 4 squared, you have 16 possibilities with 4 different types of nucleotides. And again, because there's 20 amino acids, it couldn't be 16. So the next possibility of it being 3 nucleotides would mean there's 64 possibilities of 3 nucleotides using the 4 nucleotides A, C, G, and U of RNA. And since there's 20 amino acids, 64 possibilities, that could be true. It would mean that some amino acids are coded for by more than 1 possibility. All right, so how did they figure this out? First thing they did was just in a lab make a chain of mRNA that was all U's, just U over and over and over again, so that every codon that is read by the ribosome would be U. Then they let the ribosome make polypeptides, proteins, in the test tube, and they tested it at 1 temperature, they tested at another temperature, tested at a third temperature, and it didn't seem like temperature had a big effect. Like 27 and 30, or 37 and 24 had similar effects, but at 37 degrees, a protein made from this poly-U would degrade over time, so they couldn't see it after 20 minutes. But the rate at which it was made was pretty similar to 24. So 37 degrees C is your body temperature, and that's why they chose 37 degrees C. Okay, so when they do that, they also included some experiments where there was no mRNA put into the test tube and see, do I get any protein from it? And essentially they don't get any protein from it. So what amino acids do they get when they add just U's? They get the amino acid phenylalanine. The one-letter code is F, and the three-letter code is PHE, and they get phenylalanine only when they have the poly-U sequence. So UUU as a codon must code for phenylalanine. That's the idea. UUU encodes for phenylalanine. Then they use that same approach, but now they put in an mRNA that was all A's, called poly-A. Poly means many. And again, they did it at multiple temperatures, and then they measured what amino acid comes out of this thing, and that is lysine. AAA is lysine. The three-letter code is LYS. The one-letter code is K. Do you have to remember what these codes are? No, you don't. The one that you have to know is AUG, which is methionine. So they did the same thing with poly-C. So the first three that they found were all U's, that's phenylalanine. All C's, that's proline. All A's, that's lysine. But what about some of these others? Well, they can in a test tube, you can put the RNAs together in any combination you want. So you can do UC, 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 and so on. And then they do that, they got a bunch of serines, and so on. And then they change the third letter, and so on. And this is the table that they ended up with, which is the codon table that we use today. And it's pretty much a universal codon table, no matter what kind of organism you are on the planet. If you're a bacterium or if you're a giraffe, this is the code that you use. It's a universal code. And that if there's a UUU on your mRNA in your bacterium, that codes for phenylalanine. If there's a UUU and you're a giraffe, that also codes for phenylalanine. So that's pretty convincing evidence of evolution, actually, that this evolved very early in life and has been retained throughout life. There are a couple exceptions. Mitochondria use some weird codons, and there's some other organisms that use a couple strange codons. But there are cases where some amino acids are coded for by multiple codons, like leucine can be coded 6 different ways. It could be UUA will get you leucine, or UUG, or CUU, or CUC, or CUA, or CUG. All those things will code for leucine. All of these would be different tRNAs. All of them would bring a leucine to the ribosome. Some things are unique, like AUG is the only way to code for methionine, and that's it. There's one start codon. There are several stop codons. Remember, we talked about the stop codon. At the end of translating a protein, you run into a stop codon. Oops. And that stop codon is the signal to the ribosome that it's done, and it takes the small subunit and the large subunit and breaks them up and releases the protein. Okay? So if you just had nucleotide sequence, okay, in an mRNA, you could use a table like this and figure out what is the amino acid sequence based on that. Okay? This is to say that in many cases where there are several codons coding for the same amino acid, the second codon, the second position in the codon, the second nucleotide in the codon is the same all the way through. So all those different versions of leucine, number 2 position is always uracil. All these cases of serine, the number 2 position is always cytosine. All these cases of arginine, 1, 2, 3, 4, 5, 6. The second codon is always glycine. And all these cases, here's a case in serine where it's either a C in the second position or a G in the second position. Usually the third position is the most variable. Usually that's the most variable. And sometimes that's written as like a U or a C, and then a U, and then N, meaning any of the nucleotides will do for the third position. Sometimes this is called the wobble position. So later on when we talk about mutations, it depends on where the mutation happens. It's going to typically be least damaging if it's in the third position, because it doesn't really matter. You'll still get a leucine out of it. And it tends to be most damaging